Welcome to week 12, where we're going to be talking about gene expression regulation, but mostly meiosis and sexual reproduction. So last week, we covered the central dogma of biology, where we talked about how DNA gets converted into RNA, and then there's some processing with that poly A tail and that cap, and it's that sequence of nucleotides that get read to be made into protein. But what we didn't talk at all about was how it's regulated. And how it's regulated is actually really important because we all start out as just cell, one cell, where we get half our genome from one parent and half our genome for another parent, which is everything we're going to be talking about in the rest of this presentation. But we see that we are a fertilized egg and that single cell is going to multiply and multiply and multiply, and that's what we saw last week in mitosis. But what that means is that all the cells in your body carry the same DNA. So we have our single zygote that undergoes mitosis and mitosis to develop into a blastocyst. And it's that from which you can see all the different cells. So our skin cells, our neurons, our muscle cells, our endothelial and epithelial cells, they all have the exact same DNA, and yet they're totally different looking cells that do totally different things. How is that? It's all about which genes are expressed. So we talked about genes in the last presentation, where you have these different segments of DNA that get converted into those proteins. So which genes are expressed determine what type of a cell is made. This is called cell's differentiation. So you can just see in this example, you have one type of cell, the different proteins start to get expressed, and then those cells develop into all the different types of cells in our immune cells. So what determines what's expressed? Transcription factors. So these are the proteins that are going to find that entire sequence of 6 feet of DNA within our cells. They're going to find that specific region called the promoter that they are specifically going to bind to. And this is what is critical for the initiation of transcription. So this was again last week. And just as a side note to talk about transcription and how it's regulated, when we talk all about the immune system and what's going on in our COVID reactions, really what it is it's these different transcription factors and what different signaling to make these different proteins that are going to then be able to bind and determine which pathways our immune cells go under. So you don't really need to know anything about this, but it's just to understand it's these transcription factors that sort of determine what gets made. So back to thinking about how cells become those different types of cells to do all those different functions. They are going to be expressing different proteins. And the point of this is it's all very tightly regulated. So we're not going to go into the specifics here in this course Bio 181, but know at every single one of these steps, there is a whole lot of regulation going on and of course a whole lot of research. And the primary level of regulation to see which proteins are going to be made is to determine what mRNA is going to be made. So that was that level of transcription factors. But then of course, once that RNA is made, there's a whole new huge area of research and understanding that's looking at how different RNAs regulate RNAs. So there's a whole lot of research going on there and a whole lot of controls. There's also, of course, controls in the production of the protein from that mRNA. And then, of course, there's how long proteins remain and different proteins are stable for different amounts of time. So every point, there's a lot of regulation.